Okay, hi everybody, I have 115. So let's talk first maybe about what the quiz on this week is going to cover, right? So for the quiz last week, we had that include, I think, part of chapter 3, right? 1 and 3.
If it's Magnum.
And I have here, we went as far as, or up to section 3.4 on page 75.
That's not Magnum.
Tiffany, could you mute?
Oh, sorry, I didn't realize it wasn't.
So we're going to pick up then with section 3.4 in chapter 3. And I thought we would include section 4.1 and 4.2 in chapter 4. Because I posted the the lecture Zoom on that, so you have access to that. Any questions on what parts of chapters 3 and 4 will be on lecture quiz 2?
I'm sorry, you said 4.1, 4.2?
Yes.
Okay.
4.1 and 4.2.
Okay.
So my intent then would be to have that second quiz open like we did last Thursday from 1:15 to 2:30. Several of you have talked to me about other times, so we'll stick to those agreed upon times. And that's kind of how we'll do it. Any questions on that? All right, great. For those of you that are taking lab and are in quarantine, I would like you to email me the lab reports for, I think it's 5 and 7. Some of you have done that already, scanned and emailed me those, so I appreciate that. Please get those to me by the end of the day. By that, I mean by 5. And I am contemplating sort of how we might come up with a makeup experience because the exercises we're doing this week in lab are really super critical for you to come in and actually do. They are establishing good aseptic technique transfer techniques that you've got to come in and you've got to do them because they're just critical. And I know it's by no fault of your own that you're having to be quarantined, but it is what it is, and we got to kind of work around it as best we can. Then the good thing is we will not have lab next Tuesday because of President's Day weekend. You all are aware that we're off the 15th and 16th. And I'm going to, again, be thinking about this, and I'll send an email out to those of you impacted, but my initial thinking is to maybe have Wednesday, the 17th, as a day where you could come in because I am here that day. And I have to be here while you're doing that manipulation of bacteria. And I don't know if that's going to work into your nursing schedules or how that all pans out yet. I don't know. But initial thinking was have the 17th available all day for you to come in and try to do those exercises. I'm not asking for feedback right now. If you want to send me an email, you can, but I'll be in touch with you with regard to more possible details on the makeup. Okay. How about questions on stuff? Chapter 3, Chapter 4, Chapter 1, whatever the case may be.
I have a question, like on germination. Can you like explain that a little bit more to me? I'm just not comprehending what's in the book for some reason.
On what section, Sarah?
It's section, what section is it? It's on germination.
Oh, of the
It's on page
endospores.
108.
Of the endospores.
Mm-hmm.
Yeah, okay. So let me pull up the PowerPoint here. I think that was chapter 2, no, 3, right?
No, it's chapter 4.
4, okay. Thanks. All right, that is being uploaded. Should be here in a second. All right. Okay, Sarah, where in chapter 4 was this again?
4.4.
Section 4.4?
Yes, it's on page 108 in the book.
108. Oh, gotcha. All right. So this is not something that I have, it's not in the Zoom lecture yet.
Okay.
So everybody's aware of that. I recorded up to actually this very section on internal structures, 4.4. That's where I ended. If you looked at the PowerPoint or looked at the Zoom lecture, this is where I'm going to pick up with that. I can still answer your question, certainly, but it's not something that I would have expected you to get to. But if you're there, kudos, good job. You're working ahead. That's good.
I mean, if
So I was
anybody
thinking,
else has any other questions regarding like right now's material, then go ahead. I can wait for my question next week or whatever.
oh no, it's all right. So what Sarah's asking about is the process by which the endospore is formed, or what happens to it once it encounters a suitable growing environment. And this is, again, going to be discussed in more detail in the next Zoom lecture. So you have to understand that when we talk about bacterial cells, those that we are utilizing in lab and culturing and in this week's lab transferring from broth to broth or broth to slant or plate to slant or whatever it might be, those cells are in the vegetative state. It's kind of a funny word, vegetative, but that's the word that's used. So it's the form that you and I are most familiar with. It's the form that is what we encounter in bacteria 99.999% of the time. There are some bacteria that are called Endospore Formers, and the genus Bacillus, those are all Endospore Formers. If you've ever heard of Clostridium, you've heard of C. diff, a lot of you have, right? C. diff, C stands for Clostridium, D stands for difficile, I think is the species. If you've ever heard of gas gangrene or gangrene, that's formed from Clostridium. So it is also an endospore former. And so when Bacillus or Clostridium vegetative cells encounter a decrease in nutrients, or if they encounter a harsh environmental state. Maybe it starts to dry up, maybe the temperature starts to fall. Whenever they find themselves in a less than optimal growing environment, then they will form an endospore. Each vegetative cell can form one endospore. And that's sort of what this slide begins to talk a little bit about. It goes through the different steps or stages in the formation of this resistant structure we call the endospore. There's this YouTube video that will go into some additional detail that kind of shows you an animation of the process. But in essence, the endospore is a structure that is able to survive in very, very desiccated environments or conditions. It tries to release as much water as it can, and it does so with the help of a chemical called dipicolinic acid, which you'll be again hearing about, or calcium dipicolinate, I think it is. And this particular chemical is what allows the endospore to basically get to that most dehydrated state that it can, because to an endospore, water could be problematic. So it creates this very, very dry structure. And then when conditions improve, when the temperature gets back to normal, or more commonly when nutrients become available, then we have this germination process. Again, funny word, we think of a seed, germinating into a plant. It's a different situation here, of course. The germination of the endospore results, as we can see in the upper left-hand corner of this slide, in the formation of our common vegetative state of the cell, basically. So that sort of in a nutshell is the process. If you can just wait till I get to that next section of the next lecture, we'll talk in a little more detail about the process. How about other questions on anything you've been looking at in chapter 3 or 1 even? We can talk about anything you want.
I don't have so much of a question as much as I have like a request. I was wondering if maybe we could actually start going over like the key points in like what we need to learn for that week during these sessions, just because I feel like I retain better when I'm hearing it multiple times. I know a couple other people have said the same thing. I struggle with reading and comprehending sometimes. And it helps to hear it. So I was just wondering if that would be something we could do.
We certainly can do that, absolutely. What I'm going to probably just reiterate is sort of the main sections of each chapter. Because in terms of going into detail, that's sort of what the Zoom lectures do, I hope. But if providing you with a bit of more of an overview, I can certainly do that, sure. Where would you like to begin to talk about that? Any particular chapter? You're muted, Ashley.
Yeah, I was trying to find the mute button. I was wiggling stuff around. Hopefully, like if we wanted to just start from 3.4 and then into what's going into the quiz.
Sure.
Like what will be on the quiz that Thursday, maybe. Not what will be, but like the sections. Sure.
Well, unfortunately, these quizzes, are covering a significant chunk of material, and it's only 5 points. So it's like, well, how do I study everything? So I certainly get that frustration on your end. As far as the quizzes go, I kind of waffle back and forth between not even doing quizzes But then I have students tell me, well, I really like the quizzes because it forces me to study. So I don't know, you're damned if you do and you're damned if you don't sort of thing. But basically this next section that we're going to start this quiz number 2 material with is continuing to talk about the 6 I's that were introduced earlier in the chapter, right? Those are depicted there on page 63. And they're starting the inoculation I guess, in this section 3.4. So as we have started to do in lab last week, and again, I'm sorry for some of you who are taking just lecture, not lab, because I keep going back to lab because I think they're just integrated with one another so well that it helps to be taking both at the same time, but you don't have to do that. But in lab last week, you started to grow some bacteria, and you saw the culmination of that, I hope, when you examined your tube with a swab in it. And we also did, what else did we do? You did a plate, didn't you? Yeah. So you know what growth kind of looks like, hopefully. In a broth, it's a cloudy turbidity, right? And on a plate, it's evidence of some colonies present on the surface of the agar. In lab this week, we were hopefully going to make some transfers. So when you go to make transfers, we have to adopt special types of aseptic techniques to minimize the chance of contamination. And so they talk about the importance of isolation here on page 75. Having isolated colonies is really critical if you start with a mixed culture and your goal is to separate those cells out to a point at which you have a colony forming from that isolated cell that you can then test and try to figure out the identity of that unknown bacterium, for example. So isolation, the ability to isolate, one of those 6 I's is isolation, is really, really, really critical. And they talk in here about 3 different methodologies that you can use to isolate bacteria, 2 of which we did and will do this afternoon in lab. Those being the streak plate and the pour plate techniques. Again, I don't know to what extent you want me to go into detail about that, or just again, can I continue in a general sort of way?
Yeah, just a general.
Okay. So these 3 techniques are describing ways in which you can take a mixed culture and from that separate out the individual cells to the point at which you have separate colonies. And we assume that that colony of 1 million cells came from an isolated cell that underwent rapid cell division. So that 1 cell becomes 2, the 2 become 4, the 4 become 8, 8 becomes 16, and so on and so forth. We have what's called exponential growth occur. So within a very short period of time, like within a day or 2, you can go from a plate that has nothing growing on it to something that looks like one of these 3 plates, right? You got growth. And it's really kind of fun to see. It's amazing how fast this can happen sometimes. So isolation is a really important technique to be able to achieve this process of separating out a mixed culture into individual colonies, individual species. When you get your unknown, those of you in lab in another couple months, you're going to be performing a battery of biochemical tests on that bacterium. And that's what they kind of talk about next, this ability to identify what it is we're working with. And as I said, there's a zillion different tests that you can perform. We're going to be doing probably, I don't know, 15 or 16 or more different tests. And then you basically take that data, the results of those tests, and you utilize different techniques to try to determine what exactly it is you're dealing with, you're working with. And so on page 78, they have a flow chart there. I also provide some additional flow charts on the PowerPoint Zoom recording. These are basically simple dichotomous keys, meaning you make a choice. For example, at the top there, where it talks about gram-negative cocci and coccobacilli on page 78, if it's an oxidase negative test that you got, then that indicates you probably have a Acintobacter as the unknown. If it's an oxidase positive test that you got from that unknown, then you'd have to ask yourself, well, did it ferment lactose or not? And you just kind of work your way on down this stratification until ultimately you come up with the identity. So that's one way to use tables and flowcharts to determine that. There's other techniques like the rapid mini test that I talk about in the lecture that some of you will be able to use, hopefully. And then the chapter gets into different types of media. It breaks it down into physical states. And again, if you've taken lab or are taking lab, you know what a broth is. It's a liquid. Today we work with slants, we work with plates. You had a plate last week as well, so you know what a solid media is. There's also semi-solid media, it's called, and we'll be working with that coming up in another couple months. And the particular semi-solid media that sort of is between a liquid and the solid auger is called SIM, S-I-M. It has this clot-like gelatinous consistency to it. That's kind of the best way I can describe it. It's like snotty egg white, kind of. It's kind of solid, but it's not watery. And that's sort of an interesting media. You just got to kind of have to see it to know what I'm talking about. But we don't use it a whole lot. We generally are using liquids or solid media in the lab. The solid media, again, could be a plate, like an auger plate. It could be an auger slant. An auger slant is basically where you take molten auger and you pour it into a test tube, and then you cap that tube. You put it in the autoclave to sterilize it. And then when it comes out of the autoclave, because the temperature is really, really warm, it's probably like close to the boiling point of water. You take what Deb does, our lab tech, she takes all these tubes and she lays them so that the cap of the tube is laying on a glass rod that she's taped to the table. If you come to lab this afternoon, you're going to see what I'm talking about. And as that cools, the auger cools on in a slant-like morphology or shape within the tube. It's called a slant. And that's, again, a very common solid media that we would produce and use. What makes auger auger? Well, anybody know where auger comes from? Tiffany, any idea? She should know this because she had lab today.
Red algae.
Right, red algae. Yeah,
I said it, but I wasn't unmuted.
Somebody beat you to it. Who would ever think, and how they ever discovered red algae or began to use red algae, I do not have any idea what the historical significance of that whole thing is all about. But nonetheless, there's a particular genus of red algae that is harvested and utilized and refined, and that is the material that is used in today's microlab and has been probably for 100 years as a solidifying agent. And I want to really point out the fact that auger has no nutrient value to the bacteria. I want to dispense that thought in your mind because most students think this and they get it wrong on the quiz. Auger, they don't eat the auger. The microbes don't eat the auger. The auger is there simply as a solidifying agent, helps the material solidify. We add nutrients to the auger, and that's what the microbes are metabolizing. And I think they say something like most media contain between 1 to 5% auger. But that's what that material is, auger. And then they talk a little bit more about what a chemically defined synthetic medium is versus a non-synthetic or complex medium. And I would just have you reference page 81 in your textbook, and you can kind of see vividly the difference between a chemically defined synthetic type versus a complex non-synthetic type. What's the big takeaway when you glance through those two tables, if you happen to have your book? What seems just to be real obvious?
What
Anybody?
page did you say again?
I'm sorry?
What page was that again?
Yeah.
81. 81. Table 3.6a and 3.6b.
The non-synthetic medium looks a lot easier to make.
It sure does. If I'm Deb, our lab tech, and you told me which of the two would you rather produce or make, I'm going to pick the lower one because I'm lazy. No, I'm really not lazy, but boy, if you want to make the chemically defined synthetic one on the top, I've got to weigh out, some pretty small quantities of various amino acids. I mean, my gosh, how many amino acids are listed there? 2, 4, 6, 7. There's got to be 18 amino acids in there. Holy Moses, And then look down in the additional ingredients. 0.005 moles of nicotinamide, blah, blah, blah. I'm not a lab tech, thank goodness, because I don't know how you measure a mole. I can't remember from college chemistry what a mole is, but I think it's safe to say it's not a whole lot, right? So it's very time consuming and laborious and expensive to make that kind of a medium. And you really have to have some good technique, I'm sure, behind that as well. So when I think of, say, fastidious types of bacteria, those that are picky eaters, quote unquote, I might have to use a synthetic media to grow that bug on, because it's just so specific in its needs, its nutritional needs. The bacteria that we're using in our lab and most micro labs are utilizing complex non-synthetic media because, number one, we don't use fastidious picky eaters to grow, and number two, you'd have to have a huge budget to be able to afford all the ingredients necessary for a synthetic type. And then the book talks a little bit about the difference between selective and differential media. Selective media, which we will use this semester, has particular materials in it, in the growing media, that will inhibit the growth of some bacteria but promote the growth of others. So you can sort of like eliminate certain kinds of bacteria from growing if you utilize selective media. Differential, it does what the name sort of implies. It allows you to bring out physical differences between different microorganisms. You typically look for color changes. Certain bacteria will exhibit, say, a yellow color change from a pink or a red. Others will not. I guess the most extreme example of a differential media is shown on page 83 on the right-hand side in the middle, where it says chromagar orientation at the top. And look at the names of the bacteria below there. There's 6 different bacteria that somebody has spelled out. If you were to inoculate E. coli on chromagar orientation media, it would have that pink color. If you were to have Klebsiella present, it would have this glossy dark blue color. If you had Staph saprophyticus, it would have a kind of a pinkish white color. So what they're basically saying there, of course, is you can differentiate from different bacteria based upon what sort of color you get. That's sort of an extreme example, but it's amazing. There's just hundreds of different kinds of augers that have been developed, many of which are differential, that allow you to differentiate between different kinds.
Is that how they do the culture art, like we were talking in lab?
Yeah. Sometimes that's exactly right. Exactly right. I think that pretty much hits the major topics there in the second half of chapter 3. Chapter 4 is a survey of the prokaryotic cell types. And you remember from high school bio, as well as from chapter 1 maybe, that when we talk about, life or living cells, all living cells have specific characteristics, and they talk a little bit about those in figure 4.1 of the book. This is not a figure you need to memorize or anything, but if you think about it, all cells have to be able to reproduce. All cells have some genetic material in them. In other words, they can pass on their genes to subsequent cells as the process of growth and development occur. There's materials moving in and out of cells, right? And that's kind of what they talk about here. So whether you're talking about a eukaryotic cell or a prokaryotic cell, all cells, all living cells have to have certain key characteristics. And that's kind of what's described at the very first part of chapter 4. And while I'm talking about that, let me pull up chapter 4 here. We got it. Go back. So what we're going to, in essence, do in this chapter is we're going to look at various structures found within prokaryotic cells. And we keep in mind, of course, that prokaryotes lack a membrane-bound nucleus. They lack membranous organelles. The only organelle that they share with eukaryotes is what organelle?
Ribosomes.
Right. And when we think of the taxonomy, we remember of the 3 domains, 2 contain prokaryotic cells, as depicted there on the left-hand side of the figure in front of you. The bacteria and the archaea domains contain prokaryotic cells. So in essence, we're going through initially the external structures. We then focus on the cell envelope and wrap up the chapter with internal structures common to prokaryotic cells. There's a really nice diagram here that is new to the 11th edition of the textbook. And I think they've done a really fine job with this. And I think you can always come back to this figure 4.2 on page 95. And I really like how they have here on the left-hand side, all bacteria have, a cell membrane, a chromosome, ribosomes, and an internal cytoplasm, while other bacteria may possess these other structures. So this is common to all. This is unique to some or many. And then, Sarah, there's your endospore that we know, again, they don't show this in the diagram, but that only certain types of bacteria, a very small percent really, are able to form these interesting resistant, desiccated, sort of dormant forms of certain cells, certain types of bacterial cells. So it's really then just a matter of kind of going through and familiarizing yourself with various appendages that bacteria have. And then they talk about this S layer that some cells have that can contribute to some pathogenicity. The major function of that particular structure appears to be for attachment of cells to some underlying substrate. Actually, these fimbriae that you'll learn about can also do that too. One very important characteristic of many bacteria is their ability to stick to a substrate, to some growing medium. You know, it could be in the laboratory, or it could be on the tooth of your on your incisor. You talk about plaque buildup, you know, causing dental caries. Well, yeah, that's because bacteria, which live, you know, in our mouth, some of those guys like sugars, and they like to produce acids as a byproduct of the fermentation process. And it's the acids that can eat through our enamel and cause dental cavities. But those cells have to initially be able to attach themselves to the enamel of your tooth. And so they talk a little bit about, you know, how cells can do that. Typically through the fimbriae, or from this S layer in certain types of cells. So that's basically what chapter 4 kind of does. It just takes you through a tour of the cell, basically, and talks about specific interesting characteristics and what those things do for the cell. What are the functions? Yeah, and actually, as long as we're talking about kind of an overview, the same can be said for chapter 5, only here we're focused next week on the eukaryotic cells. And we'll do something very similar. We'll take the cell, we'll divide it up into external structures, boundary structures, and then internal structures. So the book kind of carries that theme throughout the next chapter as well. And they throw in some other interesting things about you know, how we think maybe eukaryotic cells evolved. And we'll talk more about that coming up next week. So that is kind of a basic overview. The other thing that's kind of helpful too, as I'm just kind of thinking about it and looking at the chapter, at the very beginning of each chapter, they have a page that they call the "In this chapter," and they break down the various sections. For example, in chapter 4, there appears to be 7 sections, and I'm on page 90 here, if you're wondering where I'm looking. And so this might be certainly worth looking at before you start to read the chapter, just to get a flavor for what topics the chapter covers. I think that's a very, very nice way of getting a sense of what you're going to be looking at. The other thing that I will mention to you, a lot of books have this too. In the back of the chapter, I'm on page 122 and 123. They have here, in essence, sort of a, I guess I would call it they call it a chapter summary, exactly what it is. And it does a great job kind of just going over some of the major features of the chapter too. And they use boldface terms to kind of bring out those important terminology that you want to be kind of comfortable with. Those would be certainly worth looking at maybe just before the quiz, just to refresh your memory of the major topics or highlighted topics that the chapter covers. Or I guess you could even read through that first before you get into the chapter even. That wouldn't be the worst thing in the world either, to think about it in that way. I don't know, Ashley, did that kind of help give you a sense?
It did.
Good. Yeah, I'm happy to do that. Absolutely. So thank you for that comment.
I just know, like, with the last quiz, I struggled. I read everything, and then as I'm going through the quiz, I'm like, I remember reading about this, but I can't remember exactly, like, specifically what it was or how to explain it.
Yeah. Well, one of the questions was, when did the first, what was it? I forget the question now. When did the first prokaryotes first evolve on Earth? I think it was something like that. And you kind of had to know that time frame.
Yeah.
Yeah, again, I get the dilemma of 5 points over 50 pages. It's like, what do I study? Yeah. I wish I had a magic answer for you there. You just kind of have to,
Just wing it.
well, not wing it, but there's just a lot of material. I mean, you think about A&P 1, A&P 2. My gosh, think about what you learned in a span of a year. You learned a lot. You retain 5% of what you learned, maybe if you're lucky, right? That seems to be the common statistic they always give. I think the terms that are in boldface, I think those are really critical key terms. I think, that's something that you should know. As I look at those terms, as I peruse through chapter 4, those words I've used in the lectures. Those are the terms you see on the slides. I think you should use the Zoom PowerPoint lectures as a guide, obviously. Hopefully you're using those. I mean, the PowerPoint slides are really, should be, providing guidance for you as you study and learn the material. Now, again, there's a lot there, I know. But I think having had A&P 1 and A&P 2 should make this not so much of a daunting experience as if you were to have walked into micro without any biology before. Then I could see where this could be extremely challenging. How about other specific questions on any of the material? Remember, next week, not that it impacts you guys so much, I suppose, we don't have class Monday or Tuesday, as I said earlier, because of President's Day. And then as I look at the syllabus, that following week, we have our first lecture exam on the 25th. So that's like 2 weeks from Thursday, I think. So it's a ways away, but time flies.
I wasn't sure. I think it was Akira. That's the micro tutor.
Yeah.
Is she gonna post a Zoom study session or something?
I know she's meeting with my A&P students Sundays from 8 to 9. I would think she'd be happy to do something similar for you guys. So has anybody reached out to Kyra? I think I may have mentioned her name to you before. She's in the Learning Center, and I think she has some on-campus hours, but she of course has Zoom times as well. So I would send her an email. Did I give you? I can't remember what I sent you and what I sent the A&P students. Did I give you her email?
I don't have it now.
Okay.
I can message her right now and ask her if she would.
Say that again.
I said I can message her right now and ask her if she would. I have her in the nursing group right now.
Okay. I was just gonna find her email, and you guys could ask. Let's see. I think I've got it right here actually. Yeah, Kyra Nolder. I'll just put in the chat see if I can do this. So there's her email, Nolder KK01. And I suspect she'd be happy to develop a time to get together via Zoom. She's taken micro, obviously. She's taken ENP1 and ENP2. She's a very good student, and she enjoys tutoring. And then don't forget also, if you ever want to meet one-on-one with me, I have Monday office hours and Tuesday hours, and I think even Wednesday an hour. So, but I'm asking that you go through Starfish to kind of book an appointment, because then I'll get an email confirmation that says, you know, Julie wants to meet with you Monday the 22nd at 10 o'clock or whatever the case may be. Other questions on any specific parts? Chapter 3, chapter 4? Have you gotten into chapter 4 yet? Hopefully you have. Hopefully those of you in quarantine will stay healthy, I hope. I don't know if I told you I get my second COVID shot Friday. So I'm kind of excited about that.
Can you explain planktonic growth?
Yeah, the planktonic cells relate to the motile forms of cells that relate to a biofilm. So if we go to the PowerPoint slide, and there's a diagram in your book. Do you see the formation of biofilm there? Okay. We're going to be spending more time talking about biofilms in an upcoming chapter. These are just uber interesting. They are fascinating. I would love to teach a whole course just on biofilms. It's just amazing what happens here. So basically, Melissa, what they're showing here are these seemingly flagellated cells, right? That's what it looks like. They got a flagellum here. And the reason that they put a flagellum there is to denote the fact that they're motile. They're able to move. Now again, when you think of bacteria in a biofilm, they're not swimming through the air generally. They're swimming on the surface of what is likely a moist, wet environment. That could be on the surface of your tooth enamel, as shown here in the upper right-hand corner of this slide. Yeah, believe it or not, if you were to scrape your teeth, you'd find a lot of bacteria. And I talked just a moment ago about dental cavities, dental caries, right? As a result of acids produced by bacteria that live in biofilms on our teeth. Yeah, but you have biofilms all throughout your body. You have them lining your digestive tract. Another good place you can find biofilms. You can go to your kitchen sink and pull out the drain and look underneath there, and you see this kind of yellowish, tan, slippery, grimy junk that you don't even want to look at, or the bottom of your tub. Do you know what I'm talking about? Okay, I'm glad I'm not the only one that has that. Well, that's a biofilm. Yeah. Now I know it's gross. We want to get rid of it. It's like, oh my God, get the Ajax out or whatever and clean that up so I got shiny silver stainless steel, whatever. But if you were to delve into that biofilm in the sink, on the tub drain, on the rock on the edge of the shore of a lake, on your tooth, there is a whole host of, where'd my slide go? There it is. There's a whole host of bacteria, not just one species like they're kind of depicting here on this slide. There can be multiple species of bacteria and actually even other types of cells, even eukaryotic cell types like protozoa can live here too. It's just bizarre. Fungi. And they live in a mutualistic, cooperative way. And again, we'll get into more details of this coming up in another chapter. But let's just stick with bacteria for now. Let's say this is a biofilm of bacteria, and there's several, half a dozen or more species living in that scummy stuff on the drain, under the drain of our sink. They have this cooperative ability to share nutrients, to know when to turn on certain genes, when to turn off certain genes, whether that's going to be a positive thing for the colony or a negative thing for the colony. And a point will come in time when these sessile cells, the cells that aren't motile, okay, they're just kind of like hanging out, where they will change and morph into planktonic cells, which can then basically leave the biofilm and then spread to other surfaces, and you get another biofilm forming. So that's the major difference there, I guess. The planktonic cells are the motile wanderers, the cells that wander from the initial site. And this biofilm knows when to produce those planktonic cells. It is just an amazing, amazing thing to study. The book just gives you a couple paragraphs at this point, but we're going to get into more detail. We're going to talk more about this quorum sensing process, this real fascinating behavior that biofilms have. This gets at altering expression of genes. It's just amazing. I'll show you a really interesting video. It's a TED Talk. Have you guys ever heard of TED Talks?
Yeah.
Oh, they're great. I mean, this researcher from, I think she's from Yale University, she talks about her research in biofilms and quorum sensing and communication between different species of bacteria and how that can impact our health and the development of drugs. It's really interesting stuff. And to think you only asked about what were planktonic cells.
Thank you.
Other questions? Well, I will post the last half of chapter 4 Zoom lecture. I'll probably do that Thursday is my hope, so that by Friday, you can wrap that chapter up whenever you want. And then my goal will be to get the first half of 5 up. Probably work on that this coming weekend, and then finish that up, by a week from Thursday or so. Does that seem to work with you guys, as I do that, in terms of your readings and so forth? Yeah, okay. Good. Okay, again, I will stick around. I've got another half hour before my afternoon lab starts. So I'll be here. I'll answer any questions you have or chat if you have any concerns. Otherwise, I will see you guys next Tuesday and look for the quiz to be posted.
All right, see you in lab.
Okay. Carol, you're still there, aren't you? Yeah, so I will make sure we remedy the situation from last week.
Yeah, I was
Yeah.
gonna message you about that. Just
Yeah, no,
because.
I haven't forgotten about that, so you'll have
Okay.
ample time to do it.
All right, I was just concerned about the grade, but all right.
Yeah, I'll so
Figure it out.
You started at the trying to think, did you have a different time or not?
No,
No.
it
you're
was
just
at 1:15.
Yeah, and I just wanted, yeah, are you clear on the fact that the quiz is open from here to here and you've got, you know, time block somewhere in that span?
Yeah, I did. Yeah.
Yeah, sorry for that confusion.
No, it's okay. I just, yeah, the retake I wasn't able to do, so.
Yeah, now, I think there was only one question that you did not answer.
I was on the fourth one when I ran out of time, so.
I thought I looked and you had all the questions answered but one, as I remember. I can
Oh,
go
no,
back.
I was on the fourth, but it's,
Well, do not
Yeah.
worry about it.
okay.
It's one quiz of 10 or however many we have. 8, 10, drop to 8. Yeah, so it's not going to impact things significantly. So don't worry about it.
Okay. All right. All right. Is that it?
Yeah,
All right. Thanks.
sure.
So I just had a question regarding the lab makeup.
Right, and I think I emailed you back and I said I would be in contact with you guys about how we might want to consider doing that makeup. Now, I
Okay.
know I mentioned earlier that I was kind of tentatively thinking about whether the 17th would work.
Yeah, so I mean, everyone that was quarantined is in the Wednesday, Friday lab anyway. So
Uh-huh.
typically we would have lab, and next week we don't. We have simulation. So like it's just an hour block throughout the day. So I would assume that most people could make it up. But I guess how much time should we set aside? Like, are we gonna need the whole 3 hours?
No, I don't think so, because exercise 18, for example, is on culture media. And I said, read next to that exercise in the syllabus. So I took the students that came this morning over and we looked in the prep room and I kind of described to them sort of what Deb does and so on and so forth. So that whole thing would not be something you'd have to really worry about doing. No, I think you could probably get the majority of that done easily within 2 hours.
Okay.
I think what I will probably do is put together a little Zoom recording outlining sort of what I would like you to do when you guys come in, just to kind of give you a little bit more guidance.
Okay.
Because, you know, boy, you missed a really important lab. And it's not your fault. I'm not saying it's your fault. You couldn't help it. But this particular lab where we learn how to do those important steps of aseptic technique, it's really critical that you do it. You physically get in there and do it. You can't simulate it. You got to do it. It's not hard at all, but you've got to do it.
Okay.
So you're thinking if I do a Wednesday for the majority of you, that would be a possibility.
Yeah, I'm thinking that's probably a good possibility for most of us. I know I talked to Sarah, Crum, and Liz already, and they were both thinking to come in on that day.
I'm a little restricted in terms of I can't have you guys come in whenever you want because I should be physically on the floor. Now Wednesday I have A&P lab in the morning, and then a Zoom lecture with them in the afternoon, so I'm here. So that would really be a good time to do it. The only other time that you could do it might be on a Tuesday, and then we're looking at the 23rd, which we could still do. So if you were in the afternoon lab, maybe you could come to the morning lab and do the makeup. Or if you're in the morning lab, you come in the afternoon lab and do the makeup. I mean, that would be the kind of a plan B in my mind.
Yeah, I think that Wednesday would probably work for a lot of people, but so if you have lab in the morning, I guess what time would be the best time for you?
Well, I'm not going to be there to walk you through it. I'm just
Okay.
saying I have to physically be present on the floor when you do it.
Okay.
So in my mind, I could coordinate with Deb, our lab tech. She could have all the necessary materials out, and I get here like at 8:30, and I'm here until at least probably 3:30. So I'd say get in here and get it done between 8:30 and 3:30.
Okay.
I will get back to you with more information, but that's sort of my preliminary thinking.
Yeah, I would think that it probably would work for most of us.
Well, I'm kind of glad we have next Tuesday off, because it might be a good way for you guys to make it up and not be really behind the schedule at that point then.
Yeah, it kind of worked out in an all right manner.
Kinda. Yeah. As long as you don't get it again,
Yeah.
or
Yeah, whatever. Get quarantined again.
right. Right. So let
Okay.
me give thought to this, and I will put together some better thinking and thought process and get back to you guys.
Okay. Sounds good. Thank you.
Sure. And Sarah, I know you had emailed me about trying to tape it. I'm not set up to try to tape or Zoom, or I can't do that. I got a little bit of camera, and it doesn't work.
Yeah, no, it was, everybody's kind of scrambling. No one obviously wants to miss any lab. It's crucial. And I think we all understand that. It's just, this way we wouldn't be totally out of the loop with everything. I think that was the primary concern, and some of the students had even said, hey, maybe we can record it just so, we'd have an idea of what was going on versus having to come in and make it up. I'm good for Wednesday. That is my only day off. So, that's not an issue for me to go either.
Well, that's great, because I was kind of worried maybe that was going to be a problem. In all honesty, exercise, any of these exercises, 8, 9, it's just a matter of following the steps that are outlined. It's not something that and you can't do. It's not hard. It's not at all hard, but you got to follow the steps to a T.
Yeah. I mean, it's just basic inoculation stuff, right?
Yeah. It's not hard, but it's important. It's important to know when to flame, how to flame. There's little steps that you got to know in what sequence, proper sequence. That's all. Yeah.
So it's not, I've done it before. So for me, it's not.
Oh, okay.
Because I did micro in North Carolina. They just didn't transfer my credit. So I've done all that.
Well, then I'm going to make you the headmaster.
Oh, no, please don't. That was 4 years ago. I'm rusty.
Oh, come on.
But,
you remember that.
But it's not difficult. It's it's easier than baking brownies. I mean, just follow the steps.
Yeah, exactly.
As long as you understand why you need to maintain aseptic technique and all of that, all of the concepts behind it, it's fairly simple.
Right.
So it's just, I think most of us are open for Wednesdays. I normally have Tuesday nursing lab. I am part of that group, but I was sick last week and went to the Friday group and ended up getting lucky enough to be in that quarantine because I missed one day. Yeah. So for me, Tuesdays are no good during the day because I am in that nursing class normally. Yeah. But Wednesdays are my literal only open day, and I'm usually studying, but I am more hands-on. So actually being in the lab helps me understand the material I'm reading so much better. Yeah. So that works. I did want to ask you about last week's quiz because I, did abhorrently on that quiz. I'm really irritated with myself. I panicked when I saw Cox postulate, and all I could think was I could see the images in my head and I could not articulate the purpose of Cox postulate.
Yeah.
And then I spaced, I looked at that stain, and you asked what kind of stain it was. And I saw 50 stains in my head, and all I could think of at that moment was gram stain. And I'm pretty sure that was wrong. And I wanted to see, do you go over those quizzes? Because, it's important to know what you did wrong and why you did it wrong,
Right, I do understand. This is something I've been thinking about since you took that quiz, and that is, how do I provide this access to the quiz questions? And I've got to find that out. I don't know. I'm not familiar enough with Blackboard as to know what to click on to free up those questions. What I can do in the interim, however, is I can pull up the quiz here and we can take a look at it. I think, let me try here. Okay, so hang on. Okay, do you see your quiz here?
I do.
Okay, so, I'm happy to go through this. I don't know if I can go through it with 24 people. That's
Yeah.
going to be a lot of time. But I need to research that. So here's the first question in 2 of the 3 domains. You didn't get the right terminology. You got prokaryotes and eukaryotes, so I gave you that, but the terminology really, the proper term, bacteria, archaea, and eukarya.
And that I didn't go that way because bacteria are prokaryotes. So to me, that's just,
I understand, but
you
this is asking for taxonomic domain.
yeah, and that's what I
taxonomic
did
term.
wrong. Right.
So that's what you have to know, the 3 domains.
Yeah.
Okay, so anyway, I gave you that. Here's the Koch's postulate thing.
And did I
The purpose,
actually?
the purpose is simply to be able to say that pathogen A causes disease 1. That's in essence what it is. The
Okay.
heart of what Koch's postulates involve is taking a particular bacterium and assigning a particular disease to it. That's the takeaway message.
Okay. Yeah, and I'm looking at it, and I literally sat there and thought about the experiments that go along with it and completely spaced it. Although for some reason I thought I put quotations around pure samples. I didn't, so that's good.
Well, I know you had studied it because you talk about the healthy rat and all that stuff. When you start introducing spontaneous life, I'm thinking you're merging 2 different things together there.
Well, I just, it was an example of quashing that theory.
Koch,
yet another example.
Koch was not involved in that debate.
No, it was much later.
Yes.
But to me, it was just, it's
I understand.
kind of like the flat earthers.
Yeah.
The more evidence we have that we're saying, oh, look, man, rats are not coming out of piles on the floor.
Right.
The more it's becoming scientific fact at that point. Well, it is at that point.
Yeah.
But it's just like I could see the experiment. I could remember the experiments, go with it, and I could not nail it.
Right.
Frustrating.
You got this one right.
That was easy.
Here's the stain. Now, in this stain, we're looking at a dark background and a white cell.
So we're staining the plate and not the cells, and that's
No.
why it's a negative.
We're not staining the plate. We're staining a microbial sample.
Right.
And you can see the bacillus-shaped cells there in white.
Right.
But the key here is dark background, white cells. That's the dead giveaway. This is a negative stain. A positive stain would be white background, colored cells.
Right.
Now, gram stain, this can't be a gram stain because A, I don't see any purple cells. I don't see any red cells. They've got to be either purple or red to have a gram stain.
And
These
that
are-
one I just spaced. I
Yeah,
sat and
that's
thought
right.
about,
Yeah, yeah, right. Well, you got a stain name down, but I can't give it. Yeah,
the like most basic stain that everybody who hasn't even taken microbiology is
Yeah.
familiar with. And that's
I know
all
it.
I could think of.
yeah. And then you got the fifth one right.
Okay.
So it's the first quiz. I don't think people need to get all up in arms about it. You're going to drop 2, and it's 5 points. This is not going to kill you or make you a A student. You know,
Right.
so it's the first quiz.
Yeah. Yeah. I'm just
Yeah. Don't beat yourself up over it.
frustrated. This is supposed to be my easy class, and I finding myself.
Hey, microbiology. If anybody tells you this is an easy class, you know.
No, see,
Baloney.
I really enjoyed microbiology when I took it before. It was my favorite class. It was just, it's fascinating just to see this, how everything flows together. And so I was genuinely pumped that, oh, you have to take micro again. I'm like, oh, cool.
Ah,
You know, of all the classes, I mean, I
Yeah.
would much rather do microbiology than statistics or,
Yeah.
you know,
Yeah.
that, so this is supposed to be my
yeah,
junk food. This is my,
Yeah. yeah.
you know, and so to see, you know, a 2.5 out of 5, I'm sitting there going, wait a minute.
No. Don't. You're too hard on yourself. No,
Okay.
don't beat yourself up over one quiz. Don't do that. It's not worth it.
Okay.
Nope. Don't do it. Don't give it a second thought. Just continue working. You'll be fine.
Okay. Okay. Well, just email everybody. I know the nursing students that are part of your micro, we're all doing a group chat. So we're all communicating with each other about missing lab, quarantine, and so forth. So, you know, if you email any one of us or want to set up a Wednesday, literally everyone who is a nursing student will be, you know, that will text each other and everybody will know what's going on.
So you're all in the same lab group,
No,
clinical group, no?
no, but we have a chat group.
Okay.
So all of the quarantine nursing students have got
Yeah.
a chat going.
Okay, great.
And so anything you communicate to one of us, everybody's sharing. So if you
Yeah.
email somebody and say, let's do Wednesday, then we're all gonna, you know.
Well, that's wonderful. I always like to take the approach that I need to make sure I hunt everybody down and send a group message so everybody gets it. But
Right.
I know you guys are like that, which is really good.
Well, it works and it
Yeah.
helps because if we forget to do something, then
Yeah,
we can
yeah.
remind each other.
You got each other's backs, and that's really wonderful. Good
So,
for you.
Now for the labs, I need to scan and send to you. I did 1, 5, 6, and 7, just the short answers.
I don't think one. What was one? What?
It might have just been the intro to lab safety and stuff, but I did do it.
No,
That's
no,
not
no.
one of them.
No, just I think 5 and 7 is what you have to turn in.
Okay. Okay.
And you can just scan those and email them to me.
Okay.
Okay.
All right. I will do that.
All right, great, Sarah. I'll see you later.
Thank you very much.
You're welcome.
All right. Bye-bye.
Bye.